So we're going to break this up now and look at phase 2 metabolism, which are conjugation reactions. So in phase 2 metabolism, instead of using oxidases, we're using enzymes called transferases. And like the name sounds, we are transferring small polar molecules onto a drug, and we're doing it to make them more water soluble. So oxygen is not a requirement like in the previous example. And so examples of these polar molecules that we like to attach, well, there's glucuronate, we have glutathione, we like to attach a sulfate or an acetate from acetic acid, sulfuric acid, whatever. So we are transferring things onto molecules. And we call these types of reactions conjugation reactions. The product as a result is called a conjugate. So let's just draw a picture of what's going on here so you can compare it to the previous example. So if we have a drug, we want to take this drug and form a conjugate. And so the way we do that is we transfer a molecule onto there. And so the name of the enzyme that actually works here, instead of being a cytochrome P450 or CYP3A4, let's say we were going to transfer an acetyl group onto it. Well, what is the endogenous polar molecule? In many cases, this is acetyl-CoA, and we've seen acetyl-CoA before. So let's say this is S. So this is acetyl-CoA. Here's the acetyl group. So I take a drug, I add acetyl-CoA to it. Let me just draw that arrow there. And the way I'm able to do this is with an enzyme. And this enzyme is a transferase enzyme, and it's called N-acetyl transferase. Oh, I spelled that wrong. I am having major problems spelling these days. A-C-T-Y-L transferase. And in all of these enzymes that work in the system, we'll see the word transferase at the end of the enzyme name. And so as a result, I get a drug. It's bound to an acetyl group. And I have a CoA here as a byproduct. And we've seen acetyl transferases work in the past. Do you remember, DNA transcription? We had the histone acetylation. We had the HATs, right? The histone acetyl transferase. Histone acetylation. And the reason I'm bringing this up is not because you need to know it for pharmacology, but it's when you can attach things, the things you've learned in the past, it helps you remember them. And so with histone acetylation, we were attaching a acetyl group onto the histones on DNA, and the result of this was to increase gene transcription. All right, where else have we seen acetyl-CoA? Hopefully you're scratching your noggin and saying, oh, wait, wasn't acetyl-CoA in the Krebs cycle? And we took acetyl-CoA and attached it to oxaloacetate and made citrate. So we also have acetyl-CoA in the Krebs cycle. C-Y-C-L-E. The point is you should know, you should have a sense of how these are working. So going back, phase 1, we're using oxygen, and that's a big component of it. Sometimes we see this O bound to it. I should label this. Occasionally we see an OH attached to it as well to make it more polar. Here we are attaching endogenous polar molecules. So what are some examples here? We have glucuronidation. This is the most common phase 2 process that occurs. We also have sulfation and acetylation. And so before I kind of mentioned alcohol dehydrogenase was an example of a non-P450 phase 1 reaction. I wanted just to give you one example of a phase 2 reaction, and that is with acetaminophen or Tylenol. So 95% of acetaminophen or Tylenol is metabolized by phase 2 enzymes, and this is the one that forms a non-toxic metabolite. Alright, so on the next slide, what we are going to do is just look at some of these enzymes. And I want you to just have an appreciation here. You don't have to really memorize too much. The first idea here is that the CYP3A4 enzyme, if this was a graph, a pie chart of the amount of drugs available now that are metabolizable, CYP3A4 metabolizes over 50% of available drugs. One of the other common CYP enzymes that metabolizes drugs is 2D6. Now CYP2D6 we've actually mentioned in the past because it's famous for polymorphisms. And by polymorphisms that means it can do, this enzyme can do different things depending on someone's genes. And so we talked about codeine, how people were rapid metabolizers, right? And they take codeine, and if they have a really fast enzyme, they can take codeine, they can activate it, and form morphine at a rate faster than the average person. And this can be problematic, especially in children. And that's why the FDA, so this was CYP2D6, the rapid, and they actually call them ultra-rapid metabolizers. And then we have, a bunch of other enzymes that you don't really need to memorize because you'll probably forget it in, I don't know, a couple months. Phase 2 enzymes. Now what I want you to become familiar with here is just the naming of it. So if you see this name, you'll understand what is the name of the enzymes that do glucuronidation. Well, the name of the enzyme here, and this is responsible for most of phase 2 metabolism, is, you'll see it maybe abbreviated as UGT, and it's actually a UDP glucuronylyl transferase. And just see the word transferase in there and glucuronylyl, and you'll know that's glucuronidation. This is actually the most common phase 2 reaction. This is the same process that's occurring with breaking down red blood cells and the bilirubin. When we form conjugated bilirubin, this is what's going on. Now we have other enzyme names. So what is GST? Well, GST is glutathione S transferase. As the name implies, we are transferring a glutathione onto a molecule. And so that process is called glutathione conjugation. How about NAT? You should know this one. We mentioned it last time. This is N-acetyl transferase. It does acetylation. And SALT, all right, SULT, sulfotransferase. I just want you to get familiar with the names of the enzymes and how they work. And then we have a couple other ones here that I'm not going to mention right now. So hopefully this gives you a good appreciation for the phase 1 and phase 2 enzymes and how this works all together. To wrap this up, we have a couple of stop, think, and repeat questions. Just 3 here. Push pause, try to do these questions, and hopefully I'll see you at the next video. Make sure if you like it to give it a thumbs up. Take care.